Hello, Assalamu alaikum, and good evening to you guys. So, today let's look at non-Mendelian inheritance. Okay, so non-Mendelian inheritance is there is a deviation from the normal Mendelian laws Sir Gregory Mendel derived. That is when there are we came across two rules that Sir Mendel derived using his experiment on pea plants. And whenever there was deviation from these, we say that these are non-Mendelian patterns. And these non-Mendelian patterns are of six types. They could be the alleles could be complete. They could be not completely dominant or completely recessive, which means in pea plants, we had green color tall plants and short plants, and when they were heterozygous, they expressed the phenotype of tall plants. But here, the alleles are not completely dominant or completely recessive, so that heterozygous state, they would be showing a mixture of both phenotypes. So this type of inheritance pattern is called incomplete dominance and codominance. And then another scene where we have non-Mendelian inheritance is when a particular gene has more than two alleles. It's called as polyallelism, where more than two alleles are responsible for a particular phenotype. And then when a single gene produces multiple phenotypes, it's known as pleiotropy. And similarly, when two or more genes are involved in determining a particular phenotype, it's known as epistasis or polygenic inheritance. They come under epistasis or polygenic inheritance. And then there is something called gene linkage, and genes which are located in sex chromosomes are another deviation from Mendelian inheritance laws. So let's look at the first deviation, incomplete dominance. So incomplete dominance is when the phenotype expressed in the heterozygous state is a combination of both the alleles. Instead of having one dominant and one recessive character expressed by each, respectively one dominant allele and one recessive allele, we have a phenotype that is a blend of both. So for this, we have the example of 4 o'clock plant, also known as the Mirabilis jalapa. This is a red color. This plant has red flower color and white flower color. So the red flower color is considered to be more dominant. Is considered, if we consider it to be dominant, and white flower color to be, this is the flower Mirabilis jalapa. Here we have red color flowers and we have white color flowers, and at heterozygous state they show a phenotype that is neither nor white and instead show a light pink color. This is known as incomplete dominance, where the first parental generation shows two different colors, which are which are not expressed in the heterozygous state. They are only expressed in dominant at homozygous state. So the third intermediate phenotype results from flowers of the heterozygous state having less red pigment than red homozygote. So if we consider it in a Punnett square, we have the parental generation with red flower and white flower. And if we denote red flower with capitals CR alleles, and then at homozygous state, there is two CR alleles, CR and CR, and white flowers can be denoted by CW and CW. these will be separated during uh, allele formation, gamete formation, so that there will be two CR gametes, and here we have two CW gametes. And when they form the offspring, the first offspring of the F1 generation, they form an offspring with the allele combination CR and CW. So CR and CW, CR is the allele responsible for producing red color pigments, and CW is the allele responsible for not producing red color pigments. So when there is only one allele responsible for producing red color pigment, this flower expresses the color red less than the homozygous state. So this sort of a situation we call as incomplete dominance. And when we cross two such F1 generations with each other, we get all three phenotypes: red flower color, red flower color. This would be red, and this would be pink. Both of these would be pink, and this would be white. So the ratio would be red, pink, and white. So if we look at it closely, we have the genotype ratio as such, right? The genotype ratio would be CR/CR to CR/CW, and CW/CW would be 1:2:1. So this similar ratio would be observed for the phenotype as well. So this is the incomplete dominance. There we have an intermediate phenotype between the two characteristics expressed by the two different alleles. And then codominance. Codominance is where two alleles contribute equally to the phenotype at heterozygous state. So in this situation, we can consider the example of blood grouping, where we have blood group type A, and group B. This is expressed by IA, and this is expressed by IB. So homozygous A would be IA, IA. Similarly, homozygous B would be IB and IB. When we cross these two organisms, we would get IA and IB allelic combination. This combination would show the phenotype where we have a group AB. So how is this possible? If we consider a red blood cells have certain polypeptides on their surfaces, which determine which blood group they are. So if they have certain one type of polypeptide, they are considered to be A, and if they have a different type of polypeptide, they are considered to be B. So during homozygous situation, they only express this polypeptide, and during homozygous B situation, they only express this kind of a polypeptide. But during this kind of a situation where they are heterozygous, they would express both polypeptides on their surface. So this situation is known as codominance, where expression of both the alleles contribute equally to the phenotype at the heterozygous state. So this would be a a Punnett square representing the genotypic ratio and the phenotypic ratio. Again, here the genotypic ratio and the phenotypic ratio are somewhat similar, are the same. Why? Because IA, this is the parental generation where we have IA and IB, IAA and IBB, IA and IB, and they would produce gametes IA and IB, and these would form the F1 generation, IA and IB. So this would be a heterozygous state showing blood group type of IA, blood group type of AB. And then they would, when they are cross-pollinated, the F1 generation is cross-matched, they would form the offspring of the combination IAIA, IAIB, IAIB, and IBIB. So here the ratio would be, genotypic ratio would be 1:2:1. Similarly, the phenotypic ratio of the blood group A to AB to B would be also 1:2:1. So this is codominance. Then we have polyallelism. So polyallelism is the presence of multiple alleles for a single genetic locus. If we consider again blood grouping, we have a third type of, we have three different types of blood groups, A, B, and O. Here, there is a gene that is responsible for the polypeptide A, there is a gene that is responsible for polypeptide B, and in certain instances, both of these polypeptide transcribing gene is absent, so they would have the allele I, simple I. So, depending on whichever allele is present, allele combination is present, they would have the blood group phenotype. So, if we consider this, in homozygous state, IA would be blood group A. And IB would be blood group B. Simple I would be blood group O. Interestingly, this blood group O, simple I allele is recessive to both IB and IA. So we know from the previous example, IA and IB are codominant, whereas simple I is recessive to both IA and IB. So if IA is present with simple I, this is a homozygous situation, heterozygous situation. Heterozygous situation where the phenotype expressed would be blood group A. Similarly, if we have simple I allelic combination, this is also another heterozygous state where the blood group type would be B, group B. Whereas IA, IB would express both the phenotypes, so they would have form a new blood group AB. So alleles for A and B are codominant. Absence of both alleles is out in blood group O. Hence, blood group O is recessive to both blood groups A and B. If we consider it in a Punnett square, this is how it's expressed. So we have IA, capital IA, and simple I cross, and they would form F1 generation that has homozygous heterozygous capital IA and simple IA, which would express the blood group phenotype A. And then we have IB and IB, and that is homozygous blood group B and homozygous O, recessive allele O, cross match together, they would give us heterozygous blood group B. When heterozygous A and heterozygous B are crossed, they would result in all four genotypes. So the genotype ratio would be in this manner. And similarly, the phenotype ratio would be also in this manner. There we have all four blood groups, AB, A, B, and O. And then we have something called epistasis. Epistasis is a phenomenon resulting from interactions between genes of different locus. So this expression of phenotype by a gene at one locus is altered by another gene at different locus. So if we consider this in a situation, um, if we consider this to be a gene, so this particular gene, if it is responsible for certain characteristics, X, then this gene, the presence of a certain gene or absence of a certain gene here at this point would alter the expression of this gene, so that this characteristic would depend on both this gene and this gene. So this could be again divided into two as dominant epistasis and recessive epistasis. So dominant epistasis. The dominant epistasis is when dominant allele at a specific locus alters the expression of a separate gene at a different locus. So if we consider this situation, we have again an allele here that is responsible for the gene X, for expressing the specific trait X. Then another gene here, this if it expresses the dominant gene at this locus, only will it be able to express this gene. So if this is capital R, this would allow this gene to be expressed. If it was simple R, if this was simple R at this gene, then this expression, this characteristic cannot be expressed. So this can be observed in the plumage color of housefowls. Housefowls have to have the allelic combination, have the allele considered to be capital C, which is responsible for the colored pigment, and simple C would be that they would be white color. They also have another gene, another allele that is considered to be I, which is the allele responsible for the production of pigments. But here, capital I allele would suppress the expression of the pigment. So if capital I was present, then it would suppress the plumage color of the housefowl. Instead, if it was simple I, then the color would be expressed. If we consider this, there could be capital C and capital C, and this should normally show color. Capital C, simple C should also show color. Simple C, simple C would show no color, so they would be white. This is how it should have been under normal Mendelian genetics. But because of this dominant epistasis, if there was capital I in front of capital C, then it would not express the color. It would still be white. If it was capital I and simple I and capital C's, again this would still be white. Why? For this gene, this part of the gene, it is heterozygous dominant. So still this would express white color. And instead, if we have simple I, then in this situation, because this is a recessive genotype and this is a dominant coloring genotype, this fowl would be colored. The same thing is applied here, where if there is capital I or capital I simple I, then the fowl would be white colored, whereas if it was simple I, then the fowl would be colored. And then when we consider simple C, regardless of whatever genotype we have for capital I, or capital I, or simple I, it would still be white color. Why? Because this gene here is in its recessive form. So the when dominant allele at a specific locus alters the expression of a separate gene at a different locus, it's known as dominant epistasis. So this is showing the two color. This is the colored fowl, and this is the white color fowl, and this shows the crossing of two dominant a dominant double dominant house fowl, white color house fowl, with a double recessive allelic white color house fowl. So when they are crossed, they would have this type of allelic combination, which is both, which is again white. Why? Because we have the recessive capital I, which is suppressing the gene. So when we cross-match two such organisms, two such F1 offsprings, they would give us this type of a allelic combination, where capital C, capital I, and capital C, capital I, if they are in the same organism, they would give rise to this genotype. So they would give white color offspring. Similarly, wherever we see capital I, they would all be white. They would all be white. Similarly, if we consider the simple C, the recessive genotype for the coloring pigment, wherever both its recessive simple C's, both simple C's are present, or both alleles are simple recessive feature, then they would also express white color. Why? Because they do not have the alleles for the expression of color. So these instances, they do not express color. Why? Because they do not have the dominant allele for the production of colored pigment. So all of these would still show white color, and the ones that have dominant genotype for the double dominant, dominant, homozygous dominant or heterozygous dominant for the pigment, coloring pigment, and homozygous recessive for the pigment color, simple I, then they would be colored, as in this situation. So this phenotypic ratio would be 13:3, whereas the genotypic ratio would be very different. And then we have recessive epistasis. So recessive epistasis is when homozygous recessive genotype of a particular chromosomal locus alters the expression of a separate gene at a different locus. So here again, we have a similar situation where the expression of a gene at a specific locus is controlled by the uh, controlled by the separate a gene at a different locus. But instead, here the homozygous recessive genotype would mask the expression of this expression of the other characteristic. If we have a recessive gene here, then it would stop the expression of this other characteristic. So for this, we consider the example sweet pea plant or lathyrus. Lathyrus has purple color purple flower color in dominant state. We consider it to be capital A, and capital B, capital B. And it shows white color flower color in its recessive. So both A and B allele should be dominant to be colored. So capital A, capital B, capital B would be purple, while capital simple A, and simple B would be white. But capital A, capital B, simple B would be purple again. Whereas capital A, simple B would be white. Or capital A, simple B would again be white. Or simple A, and capital B would be white. So if we were to look at it once again, these are the two colors. This is white and this is purple. This is in the dominant state and this is in the recessive state. The parental generation have purple flower colors that is expressed by capital A, capital A, capital A, and the recessive phenotype is expressed by simple A, simple B, and when they are cross matched, they would give an offspring with the allelic combination capital A, simple A, and capital B, simple B, and they would express the color purple. Why? They have dominant dominant alleles at both the allelic allele site, of allele. Both the alleles are dominant. So wherever both the alleles are present as dominant, they would be considered to be um, they would be considered to be dominant. They would be considered to be purple color. And wherever they express recessive gene, even if it's just a single gene, as in this situation, that they only have simple A for one allele, simple recessive character for one allele, not this situation, where we have this kind of a situation that would give us white color. So this is recessive epistaxis, where the presence of recessive allele at a particular point of the gene, if you consider a gene, where the presence of recessive allele at this situation would alter the expression, would suppress the expression of this one. So this gene would be expressed only if there was dominant allele at this place as well. If not, this would inhibit the expression of this. So the next type of non-Mendelian inheritance type is polygenic inheritance. So polygenic inheritance is where we have cumulative expression of two or more genes. Here there is there are multiple alleles which determine the phenotype, and if one of them are there, they would be considered one unit, and if two units are present, as in if they are heterozygous dominant, then they are considered to be one unit. If they are homozygous dominant, they are considered to be two units. In such a case, since they are polyallelic, if more than one allele is present, then they are considered to be two units. And if more than two units are present, If all four are considered to be dominant, then they are considered to be four units. So this can be expressed with the aid of skin tone. So here, each gene A, B, or C contributes to one unit of darkness to the phenotype and being incompletely dominant to the other alleles. So they are incompletely dominant to simple A, simple B, or simple C. So as you guys might remember, from the previous example, previous category, incomplete dominance, if the allele A is present, homozygous allele A and homozygous dominant A, capital A, and homozygous recessive capital A, simple A, they are two individual characters, and if both of them are present together, they are expressed in the dominant allelic phenotype, but in a reduced form. So similarly, here, if we consider either of the genes, capital A, capital B, or capital C, to be a simple single unit of darkness, then the presence of this gene would be one unit of darkness, and this gene would be two units of darkness. So the allelic combination capital B, capital B, capital C would be very dark skin, and simple B, and simple C would give us very light skin. And capital B, simple B, and capital C, simple C would give an intermediate color skin tone. So this kind of a situation is expressed in this kind of a chart. So we are here. We could have we have we could have the parental phenotype as capital A, capital B, and capital A, capital B of the other parental phenotype would result in a very dark skin. And if one parent has capital A, capital B, and another parent has simple A, simple B, then the skin tone would be slightly lighter because of the combination of the alleles. Whereas if both the parents have simple A, simple AB, then they would have lighter shade, simple A, and simple B, simple B. So genotype, the phenotype would vary with the genotype. So these kind of cumulative expressions, there are more than two alleles are expressed in a cumulative fashion, can be observed for various characteristics such as height, skin color, intelligence, quotient, and so many other features can be inherited through polygenic inheritance. And most of these polygenic inheritance would show a normal distribution curve. So normal distribution curve means they would have the both the double dominant and the double recessive, or in this situation, the triple dominant and triple recessive conditions are very rare. If we consider a graph of the population with respect to let's say, the height, extremely short people and extremely tall people would be rare. And the number of people who are intermediate would be high. Why? The chances of that are intermediate is more possible than having these two extreme cases. So this can be considered in this situation as well, where the presence of capital A, capital B, capital double capital A, double capital B, and double simple A, double simple B is much rarer when compared to the other types of genotypes. So these would give a normal distribution curve where the middle value is the most common uh, type of phenotype expressed, observable in the population. And then we have genetic linkage. Genetic linkage is where we have genes that are located closer on the same chromosome that would escape crossing over and independent assortments during meiosis and inherit together. So again, during Mendel's law, we would have studied that for Mendel's law to be valid, we need two rules specifically, that the gene should either be very far apart on the same chromosome, or on they should be located on two different chromosomes. So what happens if they are closely related on the same chromosome? They might escape being crossed over. So this type of deviation from Mendel's law of independent assortment was first observed in Drosophila. So Drosophila is a type of wild type of dragon bee, dragonfly. So this dragonfly normally exists in black color, with a black color body, and normal wings. But due to a mutation, they can appear as gray color body and with vestigial wings. So in this is inherited through autosomal genes. If we were to give according to normal, if it was according to normal Mendelian laws, if we were to give capital B for black color and capital B would be black, and if we were to express being normal wing as capital N, sorry, let me rephrase that. So if the body color was black, that would be expressed by the capital B allele, and if it was gray, then it would be expressed by simple B. Similarly, if it was normal wing, then it would be expressed by capital N, and vestigial wing would be expressed by simple N. So in case of a normal Mendelian inheritance, what is expected of a double dominant black body, normal winged Normal being the dragonfly, then it's crossed with the double recessive uh, dragonfly, the outcome would be something like this. They could have the allele combination like this. This is the parental genotype, and the gamete would be capital B, capital P, capital N, and the gamete from this recessive allele carrying dragonfly would be simple B, simple N, and they should cross over to give capital B, simple B, capital N, simple N. So all of these F1 generation would express black color with normal wings. And two such when two such first Pilar generation offsprings of crossmatch, crossbred, they would give the offspring these types of allele combination would be there. Gamete would be formed. So they would form the offspring types such as so the genotype would be something like this. And so on and so forth, so that our ratio would be something like black normal winged ones and black vestigial winged ones and gray vestigial winged ones, gray normal winged ones and vestigial gray vestigial winged ones. They would be in the ratio of 3:3:1, 9:3:3:1. This should have been the normal ratio. If it was if this cross crossbreeding occurred according to the normal Mendelian law. But since when the actual practical was held out, the observed phenotype was slightly different. There we had gray color, with body with normal wings, vestigial wings to have 1:1 ratio. So why did this occur? Because these two genotypes were close by, where they had gray body. Uh, Sorry, I think I made a mistake where when I expressed gray body was the normal one, and they had normal wings. Gray was the normal color, and black was the mutation. So, in this situation, the presence of capital G, or if we were to go according to my example, then they have uh, capital B, and simple N. They were split into. They did not separate. So whenever there was a capital B, there was always a capital N. The presence of this gamete was absent. Similarly, when there was simple B, there was always a simple N. The presence of simple B and capital N allele was also absent. So there were only two types of alleles, either capital B, capital N, or simple B, simple N. So this resulted in only three types of phenotypes, either capital B, capital B, capital N, capital N, or capital B, simple B, capital N, simple N, or simple B, simple B, simple N, simple N. So this only gave three phenotypes. So why did this happen? Because this these two genes, the two genes that are the gene that is responsible for the body color and the gene that is responsible for the wing size, were linked together because they were close on the same chromosome. That led to a different outcome than the Mendelian ratio. Why? Because they were placed closely on the same chromosome. And then, since I made a small mistake, the gray body and normal wings is the common one, and double the mutation is black body and vestigial wings. So when you're presented with questions, you can try. You can be careful with that. And then another interesting deviation is human sex determination. Humans have 23 pairs of chromosomes. So out of them, 22 autosomal chromosomes are there, and a pair of sex chromosomes are there. And types of sex chromosomes expressing male traits are named Y chromosome, and the others as X chromosome. So males have XY chromosomal combination, and females have XX chromosomal combination. So how does this work in normal? Normally, what happens is the maternal genes contain XX chromosomes and paternal genes have XY chromosomes, and the maternal genes would separate to give X gametes, and the paternal genes would separate to give one X gamete and one Y gamete. So when this X and this X combines, they would give XX genotype, the F1 generation. This would give the offspring female, whereas when X gamete and the Y gamete, if they join, then they would give the phenotype male. The, the probability of the offspring being female or male is 50-50. This is an interesting creation of the God where we have the only one single chromosome that is different, that would give the variation. So because the Y chromosome is slightly different to the X chromosome, they are not completely homologous to each other. X and Y are not completely homologous. Whereas if we consider two X chromosomes, they are completely homologous. So why is this significant? Because Y chromosome lacks certain genes on the X chromosome, and this um, if we were to consider this in a perspective of diseases, if the allele for the if we consider a female, they would both have the allele, and if one allele carries the disease, if one allele carries the if one allele carries the if one allele carries the if we consider a female, if one allele carries the gene for the disease and the other allele carries the does not carry the allele for the disease, then the chance of the disease being expressed is dependent on whether this gene is dominant or recessive, is expressed in dominant conditions or recessive conditions. Whereas in males, if they have the gene in the in one in if they have the disease, since they don't have the homologous allele in the Y gene, if the X chromosome contains the disease gene, they would be expressing the disease, and if they do not contain the disease gene, then they would be healthy. So this would increase the chances of this offspring having the disease. So this can be observed in red-green color blindness and hemophilia. Um, these two are recessive X-linked recessive disorders. So since they are recessive but X-linked, they are expressed more in men than in women because of the presence of them. So if we consider a situation where we have, if we consider X capital H to be a healthy gene and a simple H to be a disease carrying gene, then this would be a heterozygous female. Heterozygous female. Similarly, a capital H gene with a Y gene would result in a healthy male. So this type of heterozygous female are also called as carrier female. So since they have one disease gene, but they do not have the disease themselves, they only carry the gene for the disease. So they are considered to be a carrier. And these people, when they have offspring, they would give the allelic combination as such. This would have the they would have the gametes as this. So this would be something like this. So when the healthy gene from the mother and the healthy gene from the father are combined, there would be one female who is completely healthy. So this type of female is considered to be completely healthy. And then the healthy gene from the mother and the normal white gene from the father is combined again. But when the disease gene from the mother is combined with a carrier female, so this is a carrier. This is a female. Why? Because they have both X chromosomes, and one of the X chromosomes carries a healthy gene, and one of the X chromosomes has a simple disease gene. So even though they have the disease gene, because it's a recessive character, they do not express the disease themselves, but they are a carrier. Why? Because they could transmit this to the next generation. In this situation, the simple H from the disease gene from the mother could combine with the father's Y gene, and they would be expressed as a diseased male. So the male offspring would express the disease condition. Why? Simply because they only have one gene that is disease. So if a female has to be diseased, then they should have both the alleles as disease, which means the allele from the mother and the allele from the father both should be diseased. So the chances of the chances of such an offspring, the chances of a female getting such an offspring occurring is very less compared to that of a male having this kind of a situation. So this is how we can get red-green color blindness or hemophilia. And then we have something called pleiotropy. Pleiotropy is the expression of a single gene that affects the expression of multiple traits which are not related to each other. So by altering the gene at one place, say this gene is altered, so this would lead to multiple different characteristics of a human. So that kind of a situation is considered to be pleiotropic. So this can be observed in sickle cell diseases. Sickle cell disease is a disease where the hemoglobin is or the red blood cells is altered into a sickle cell shape during hypoxic state. So these are normal red blood cells, and due to an alteration in the hemoglobin protein of red blood cells, certain cells are produced in a sickle shape. So they would be in somewhat this kind of a shape. The normal hemoglobin would be biconvex, and these would be sickle shape. So this would be something like um, the normal hemoglobin would be capital R, capital R. This is a complete hemoglobin, completely healthy person. This is a heterozygous healthy person. So these type of people would have normal hemoglobin as well as sickle cell shape hemoglobin. Sorry, normal red blood cells as well as sickle shape red blood cells as well. And then we have simpler, where they only have sickle cell shape red blood cells. So this kind of situation is uh, fatal, whereas this kind of a situation, these kind of people could live until they are exposed to a extreme condition. So this is one such disease where we have pleiotropy. Pleiotropy is when we have one gene, the alteration in one gene affecting multiple traits. So because of this sickle cell, there would be multiple problems in the body. Similarly, we have cystic fibrosis. The cystic fibrosis is a condition causing thicker and stickier mucus than its normal nature. So because of this thicker, stickier mucus, and this happens because of excess chlorine secretion of defected chlorine channels of the plasma membrane. This again is a defect of cystic fibrosis transmembrane regulator. This is the name of the gene. So this mutation of this gene would lead to producing thicker and stickier mucus. So this thicker and stickier mucus can get accumulated in our lungs, and that would cause asthma. And this could get accumulated in the kidneys or other major organs, and that could cause major organ failure and so many other complications could occur because of this mucus, thicker and stickier mucus, even though the only gene affected is the CFTR gene. This kind of situation is what we call as pleiotropy. And then we have something called epigenetics. So epigenetics is the occurrence of a certain phenotypes of a certain characters controlled by factors than their DNA sequence or genetic code. So we know the expression of a gene is controlled by the nucleotide basis. So whether they have A gene, G, this would code for a specific amino acid, and this would code for another type of amino acid, and this would code for another amino acid, and so on and so forth. But epigenetics is where we have the same nucleotide sequence or the same DNA sequence, but their function is altered because of the methylation or demethylation of the nucleotide. So we know a nucleotide has a pentose sugar group, a nitrogenous base, and a phosphate group. So because of methylation or addition of a methyl group or removal of an existing methyl group could alter the function of this DNA sequence. So again, this alteration by adding addition of methyl group or the removal of this methyl group could be either inherited from the parent as a signal or could be due to factors from the environment. So this kind of signal, if it is inherited from the parent, then they are known as epigenetic inheritance. And then this kind of situation is observed. First, they were first able to observe in twins, where they both have the same genetic combination, but due to Methylation or demethylation at a specific point of one twin, only one twin would express this disease, schizophrenia. So schizophrenia is, you might have studied in the other lessons, is a distorted perception of reality where they have imaginary situations, where they imagine harmful imaginary environment. So this kind of situation occurs only in one twin, even though they both in identical twins, only one twin is experiences schizophrenia, and that is because of this epigenetic inheritance. So that is about non-Mendelian inheritance. So let's go through some questions. Let's go through some questions to see if we can, if you have understood this. Thank you. If you guys have any questions, you can post them now. So in this situation, in the ABO blood group system, which alleles are codominant? So if you guys can drop your answers to the, so IA and IB alleles are codominant, whereas IO is recessive to both IA and IB. So this is a polyallelism situation, and these two are codominant factors. So if you have any questions, you can post them right now. If you guys don't have any questions, let's end the meeting here. Jazakallah khair, and thank you everyone for joining.